For the last part of the DNA notes, we're going to talk about how DNA's structure allows it to function. Structure and function matching each other is a common theme we see in biology. For example, we talked about how the structure of a cell and what nucleotides it had very much correlated to what its job was. We see that DNA is no different. Its structure allows it to function. One of the most crucial ways in which that's true is the sequence of the base pairs. So when we first discovered DNA was the molecule of heredity, a lot of people had difficulty believing it because they thought it was too simple. With only 4 bases, A, C, and T, and G, they couldn't see how those bases could code for all of the complex information that heredity holds. Protein, which is more complex, seemed the more likely candidate. But it doesn't seem that unusual when you consider computer code. So binary code, which is what we use for computers, is nothing but ones and zeros. And with all those ones and zeros, we can make a computer do just about anything we want. In our next section, where we talk about how DNA gets turned into proteins, we'll use an analogy of letters to go into words. CAT is a very different word than TAC because the letters are in a different order. Similarly, AACCGGT is a very different code than AAGGTTT. So the base pair sequence is fundamental to DNA's working. Your base pairing is very similar to that of other humans, but it's unique, and though it's similar, it is unique to you, and that's what makes you you. And also how we can identify people by their DNA. The next structural component in which DNA's structure allows it to function is complementary base pairing. In Watson-Crick's article for Nature, when they first proposed the structure of DNA, they had a very short sentence saying something about how the structure of DNA would allow it to be a copying mechanism. Essentially what they're saying is because A only pairs with T and C only pairs with G, as long as we have one side of the DNA molecule, we can always figure out the other side. That's how replication works. Not only does that allow us to help us replicate DNA, or in other words, to make a copy of the DNA, it also allows us to copy just one portion of the DNA, like a recipe for a protein. Because remember, DNA codes for proteins, and so just like we can copy the entire thing, we can copy just one part of that and use it to make a protein out of it. So the next component of DNA structure that allows it to function is really more of allowing it to logistically fit in our cells. Remember that in a single cell, there's over 6 feet of DNA. So if that were uncoiled, it would be really, really long. Like I said, 6 feet. But by coiling it up, we can fit it into the cell. Of course, coiling it up isn't the only thing we can do, because even then it would still be way, way, way long. But by wrapping it around proteins and then coiling that up, it helps it to fit. So just the idea that it's helical is a big structural component to making DNA work. Finally, the bonds that hold DNA together are also very important to DNA's function. Firstly, the 2 strands are held together by hydrogen bonds. If you think about what we learned about bonds in our chemistry unit, hydrogen bonds really aren't bonds. They're attractions, and they're very weak attractions at that. Each individual hydrogen bond can very easily be pulled apart. However, when there's a lot of hydrogen bonds, those hydrogen bonds kind of team up and make it very, very sturdy. That is crucial for DNA's functioning. So remember, the hydrogen bonds sit in between the base pairs. G and C have 3 hydrogen bonds, A and T have 2. Altogether, they're very strong. So remember, lots and lots and lots of base pairs means lots and lots of hydrogen bonds. And that ensures that just while doing their general thing, they're not just going to randomly come apart like really loose magnets that fall off the fridge if you walk by too quickly. However, the idea that each individual hydrogen bond is very weak is very important to DNA's functioning. In order to use DNA for anything, whether we're making a copy of it or using it to make proteins, we have to pull apart the strands in order to expose the bases. And by having that be held together with hydrogen bonds, it makes it much easier for an enzyme like helicase to get in there and slowly peel it apart and expose one base at a time. just a side note then, because C and G has 3 bonds and A and T only have 2, guanine and cytosine bond is a much tougher bond to break apart. Lastly, if you recall that the backbone of DNA is made up of sugar and phosphate, and that's held together with a very strong bond called a phosphodiester link. In other words, a bond between the sugar and the phosphate. Those bonds are covalent bonds, which is much, much stronger than a hydrogen bond. And those covalent bonds are really going to be tough to pull apart. And what that means is that it's going to be very difficult for DNA ever to fall apart. And as long as that happens, the DNA sequence, remember the order of the base pairs, is going to remain intact. And remember, everything about DNA is all about that code. What is the sequence of the base pairs? And so by having that be a really sturdy bond, that ensures that that sequence maintains its integrity, or in other words, never changes. So just to recap, here we have hydrogen bonds. This would be adenine because there are 2 bonds. This would be guanine because there are 3 bonds. On the side, though, where we have a very strong covalent bond between the sugar and the phosphate, making sure that sequence never gets messed up. Okay, that's it. Make sure you see me if you have any questions.